Assalamu alaikum warahmatullah. Today we're going to complete part 2 of lecture 5 regarding viral infections of the respiratory system, and our lecture for today is going to be about coronaviridea. So we're going to talk about the coronaviruses or the seasonal coronaviruses which cause common cold, and then we're going to touch on the newly emerging coronaviruses which causes severe infections in humans. To start with, coronaviridea is an RNA virus. It has a helical capsid, it is enveloped, and it possesses a positive single-stranded RNA. And this is an electron microscopy picture of the coronavirus. And as you can see, the coronavirus is an enveloped virus, and the protruding glycoprotein give it this shape, which is part of the nomenclature of the virus is all about, it resembles the solar corona. Another reason why the coronavirus is called as such is that the glycoprotein, the S glycoprotein, has a crown shape. This S glycoprotein has a crown shape, so that's another reason why it is called the coronavirus. This is the structure of the coronavirus, and as we can see, it has an S glycoprotein. Also, it has an M protein which is embedded within the envelope. Also, it has a hemagglutinin esterase dimer, which is the HE that you can see here. The HE is also within the envelope of the virus. As you can see, the genome of the virus is helical genome, which is surrounded by an envelope, and that's why it has this circular shape. The genome is a single-stranded, linear, non-segmented, positive sense RNA. It is being considered as one of the largest RNA viruses, and it has an 80 to 160 nanometer in diameter, and it has a large genome which ranges in size from 27 kb to 34 kb in length. The envelope of the coronavirus is unique in that we know that envelopes are usually acquired from the cellular membranes. In the coronavirus, the envelope is acquired from the endoplasmic reticulum and Golgi during the replication of the virus inside the cell. The coronaviruses are divided into 4 groups: alpha coronavirus, beta coronavirus, gamma coronavirus, and delta coronavirus, and they all infect animals. The alpha corona and beta coronaviruses infect both animals and the humans, but the human coronaviruses are mainly seen in beta coronavirus, and another 2 human coronaviruses are seen in alpha coronavirus. We have in the alpha coronavirus the 229E and the NL63 human coronaviruses. Also, we have the OC43 and the HUK1, which are another 2 coronaviruses which are seen in the beta coronavirus group. Those 4 are responsible for the seasonal common colds that we see every winter months. And newly emerging coronaviruses like the SARS-CoV, the MERS-CoV, and the SARS-CoV-2, which is responsible for the recent outbreak and the pandemic of COVID-19. So in all, we have 7 coronaviruses that might infect humans. When did the coronaviruses first emerge? The 229E and the OC43 has emerged first and been identified and isolated in the 1960s. After that, they were the only genotypes known until 2003, when the SARS-CoV has hit in China. Then after that, 2004 and 2005, the other 2 common cold viruses, coronaviruses NL63 and HUK1, has been isolated. 2012, the MERS-CoV has been isolated in Saudi Arabia, and in 2019, the recent outbreak and pandemic of the COVID-19, and they found that the virus in the COVID-19 virus has a structure which is similar to the SARS-CoV, so it was called SARS-CoV-2. So we have four major antigenic groups: alpha, beta, delta, and gamma. Delta and gamma coronaviruses, they do not infect humans. They infect mostly birds and pigs. The alpha coronavirus can infect bat, canines, felines, porcine coronaviruses. They infect humans in addition to infecting animals such as bats, cats, dogs, and pigs. The beta coronavirus also infect bats, cows, pigs, rats, mice, in addition to humans. 5 of the 7 human coronaviruses lie within the beta coronavirus, including the 3 newly emerged infections or viruses which were responsible for the epidemics, such as the SARS-CoV, which is responsible for SARS, the MERS-CoV, which is responsible for MERS, and the SARS-CoV-2, which is responsible for the COVID-19 recent outbreak. The replication cycle or the replication strategy for coronavirus mimic other RNA positive sense viruses, in a sense that the virus needs to attach its glycoprotein, the S receptor, to the glycoprotein of the virus, the S glycoprotein, to the receptors on target cell, which are the DDP4. And after that, the virus can be internalized via either the receptor-mediated endocytosis or the fusion mechanism. After that, once the virus enters into the cell, it uncoats into the cytoplasm, releasing the positive sense RNA genome. Then the positive sense RNA genome can go directly for transcription and replication. It's a positive sense, so it can produce the proteins of the virus, and then those proteins go to the rough endoplasmic reticulum, where they assemble there to start the assembly process of the virus, and then these proteins, the structural and non-structural proteins, go through the Golgi apparatus, and together with the newly formed genome, they are assembled into the endocytic vesicle of the Golgi, and then assembly occurs, and then the virus exits the cell through exocytosis, and the virus is released after that. The coronaviruses are characterized by high genetic variability and the ability to evolute their own genomes, and they acquired that by having the ability to introduce deletion mutations within their genome once they are replicating. And it's an RNA virus. It replicates in the cytoplasm, so replication occurs via the RNA-dependent RNA polymerase of the virus. So there's a high chance of deletion mutation, and there is a high chance of recombination during replication, which is unusual for an RNA virus with unsegmented genome to occur. It's not as easy as in the segmented genome, such as the influenza virus. So those two genetic variation mechanisms make the coronavirus has the ability to introduce new mutations, and these new mutations might lead to the emergence of new human coronaviruses, or even these mutations, when introduced into animal coronaviruses, there might lead to the emergence of a new strain which might has the ability to infect both animals and the humans, and can jump from animals to humans, as we've seen in SARS, MERS, and COVID-19. What do coronaviruses do? Coronaviruses, the 229E, OC43, and the HUK1 and the NL63, they cause common cold, which is characterized by sore throat, runny nose, coughing, sneezing. They might also be able to cause bronchitis, and in severe cases, in the newly emerged coronaviruses, which jumped across species, we've seen that a small percentage of infection with these viruses might lead to development of pneumonia and is associated with a mortality rate in small percentage of those infected. The incubation period for the common cold viruses, is about 3 days. It ranges from 1 to 7 days, but we're talking about an incubation period average of 3 days. The coronavirus causes 15 to 30% of the respiratory illnesses in the common cold, or the upper respiratory tract infection in common cold. And antibodies appear early in childhood and are found in 90% of adults. So by reaching adulthood, or let's say after 15 years of age, 18 years of age, 90% of the population would have been infected with coronaviruses, and here we're talking about the common cold coronaviruses. Also, they found that the coronavirus can infect within the GI tract, and it might lead to gastroenteritis, diarrhea, vomiting. We're talking about here about the common cold viruses, and a subset of the newly emerged coronaviruses might also lead to diarrhea, and they might become infectious as a result of unsanitary conditions in the public toilets. But this route of transmission is not a major route of transmission. So most illnesses with coronaviruses are mild illnesses. Severe infections with coronaviruses were seen with the SARS-CoV, which emerged first in 2003 in China and had a mortality rate of 9.6. Also, we've seen another more virulent strain of the virus which jumped across species, which was the MERS-CoV, which the first case emerged in 2012 in Saudi Arabia, and it had a very high mortality rate. We're talking about 34.4%. And the newly emerged coronavirus, the COVID-19, which causes COVID-19, is the SARS coronavirus 2, has emerged in China in December 2019. And up to now, the mortality rate, we're talking about 3.4 to 4% mortality rate. This is the mortality rate based on the symptomatic patients and who presented to the healthcare system. If we look at most of the infections with viruses, as we know, they occur as subclinical infection. I think the SARS-CoV-2 or the COVID-19 virus does not deviate from this rule, and it causes infection in the general population. Most of these infections, let's say 80% of these infections, might be mild or asymptomatic. We're talking about only about 10 to 20% of those which has symptoms, and we're talking about 5 to 7% of those symptoms might be severe. So if we take all these facts into consideration, the mortality rate, the number of people infected might be higher than the current figures, which is around 2020. It might be in millions. So if we take this in consideration, then the mortality rate might come down to as 10% of this actual figure. It is still high compared to other viruses which seen in the winter months, such as influenza virus, which the mortality rate is about 0.1%. That's 1 in 1,000 infected with the influenza virus might become or might die of the viral infection. Laboratory diagnosis for coronaviruses. Coronavirus, we should take in consideration first the time of the year, the clinical picture, the examination of the patient, and we can very rarely go to such laboratory investigations such as direct detection, PCR, isolation in cell culture, or even serology. Unless we're talking about a new emerged virus such as the COVID-19, then the gold standard would be in this case is the reverse real-time PCR, because we know the PCR amplifies DNA. So this virus has an RNA, so we need a reverse real-time PCR in order to diagnose these cases. What about the treatment and prophylaxis for the common cold viruses? There is no specific antiviral medication for treatment for coronavirus infections, and a person needs to be well hydrated. Paracetamol may be given for symptoms such as pain or fever. Antibiotics do not work against coronaviruses, so they are reserved for severe cases with suspicion of bacterial superinfection. In severe coronavirus infections, such as seen by the SARS or the MERS or the COVID-19, we might need to use supportive treatment measures like oxygen therapy and life support, and put them in the ICU and intubate them to keep them well oxygenated. It's advised not to use ibuprofen. Recent reports has talked about the relation between ibuprofen and coronavirus infection, and as we'll talk in a moment, the ibuprofen was linked to worsening of the symptoms and increase in the mortality rate with those who took ibuprofen to treat symptoms such as pain and fever in the case of COVID-19. So please stick to paracetamol. There is currently no vaccine to treat or protect against coronavirus, and giving the flu vaccine does not really protect against the coronavirus. Here, a comparison between the newly emerged coronaviruses, which jumped across species from animals to humans. This table shows a comparison between these three different coronaviruses. So the SARS coronavirus has emerged in 2003, the MERS in 2012, and the COVID-19 in 2019. Both of those, the SARS and the COVID-19, has originated in China. The MERS has originated in Saudi Arabia. All these viruses has the bat as the initial animal host, and then in the SARS, the virus has spread from bats to the civets. In the MERS, it has been transmitted from bats to camels, and it has lived with camels for years, and then a mutation has been introduced within the camels, which made the virus jump from camels to humans in Saudi Arabia. And for the COVID-19, it started in bats, and then at the beginning it was said that it was transmitted through the Chinese cobra, but they found that the pangolin or the anteater is the animal from which the virus has jumped to infect humans. The SARS and the COVID-19 has the same receptor, which is the angiotensin converting enzyme 2, and the MERS has the DPP4 as a receptor. All of these viruses have one day or 1 to 2 days to 2 weeks of an incubation period. And for the COVID-19, which has been studied extensively nowadays, they found that the average incubation period for most patients was around 5.1 days. In the SARS, the mortality rate was around 10%, 9.6%. In the MERS, it was around 35%. And in the COVID-19, till now, today, the 19th of March 2020, we're talking the mortality rate is still around 4%. And the there are still newly emerging cases, and the fatalities are on the rise. Hopefully, the percentage of the mortality rate does not go above 4%. This is another comparison between the MERS and the SARS and the COVID-19. Here we have the MERS virus. The MERS virus, the average age was 56. The male to female ratio was 3.3 to 1 in females. The number of confirmed cases around 2,500. The case fatality rate was around 37%, and around 10% was seen in healthcare workers, and it was detected in Jeddah in Saudi Arabia in 2012. Here we're talking about the SARS virus, which emerged in November 2002, late 2002, beginning of 2003 in China, and the average age was around 40 years. The male to female ratio was 1 to 1.25 more in the females. 8,096 confirmed cases. Mortality in rates 10% was seen in 744 persons, and the infection was high among the healthcare workers. We're talking about 23%. Symptoms in SARS included fever, most common presentation, followed by dry cough and dyspnea, and some 25% of the patient had diarrhea, while in the MERS it was fever, dry cough, and dyspnea. The same presentation was seen also in the COVID-19, fever in 98%, dry cough 76%, dyspnea in 55%, with other symptoms, common cold-like symptoms. For the COVID-19, the average age was 50, and the age range was from 18 to 95. For the COVID-19, we can see that young children and young adults up to 18 to 20 years are protected, and if they get infected by the virus, it is characterized by very mild symptoms, which needs no further medical intervention. The male to female ratio was found 2.7 to 1 for the COVID-19. The number of confirmed cases has risen dramatically over the past few days, and it has reached a figure of around 200 and more than 225,000 cases, and the case fatality rate is around 4%, with more than 6,400 dead. It was first detected in December 2019 in the city of Wuhan in China, and the number of those who recovered until now exceeds 85,000 cases. Mode of transmission of COVID-19 is primarily through the droplets, which contain the virus, during coughing and sneezing, and they can spread up to 1 meter. So the safe distance is considered to prevent transmission is around 2 meters. The main mode of transmission is not through the inhalation of respiratory droplets, though recent scientific research and articles has been published that say that this could have been a mode of transmission, and it helped in spreading the coronavirus fastly, but it's still thought that the direct mode of transmission is through getting into contact with those droplets on the surfaces and then touching our eyes, nose, mouth, or face, and this is still the major mode of transmission thought about nowadays. Virus isolated in stool, also it can be isolated in stool, even for the COVID-19, but still this is a very minor role. It has played a very minor role in spreading the infection. COVID-19 has started in animals, as we said, bats and pangolins, and then jumped to humans. And one report has reported infection from humans to animals in Hong Kong, where a man was infected with the COVID-19 virus, and then he transmitted the infection into his dog, but that was a single and isolated incident. Still further investigation need to be done to see if humans can infect animals as well. The virus can survive on hands for 15 to 30 minutes. So the best method of preventing transmission is frequent hand washing for 20 to 30 seconds with soap and water. The virus can be recent publications came out saying that the virus can still or remain airborne for 3 hours, and it can survive on surfaces up to 2 to 3 days, especially on plastic and stainless steel surfaces, and these need to be disinfected frequently and avoid touching surfaces in contaminated or high risk areas. What about the infectivity and the pathogenesis of the COVID-19 or the SARS coronavirus 2? When are the patients most infectious? Patients might be infectious during the incubation period where symptoms did not develop. Also, asymptomatic persons who become infected and they do not develop symptoms, those in subclinical infection, can also shed the virus. But maximum virus shedding and maximum infectiosity is maximum patients exhibiting symptoms. So those who have symptoms are the most contagious and infectious to others. Virus shedding may occur after resolution of symptoms as well, and in a small minority of those who are recovering from and after the disappearance of symptoms, they might still shed viruses. But it's the it's the 1 to 2 days before symptoms appear and once the patient is symptomatic when the patient is mostly contagious to others. So what occurs once the virus reaches the lower respiratory tract? In the lower respiratory tract, the virus uses the ACE2 receptor on the cells, and which is most abundant in the type 2 alveolar cells of the lung. It attaches to these receptors and enter into the cell. After that, the spike glycoprotein connect to the ACE2 inhibitor and enter into the cell, replicate, and spread the infection to the neighboring cells in the lower respiratory tract. The ACE2 might also be the path for the virus to assault the heart, causing acute cardiac injury. So it can target the heart muscle, and that might partially explain why those with pre-existing cardiovascular conditions have the worst prognosis and the most mortalities are recorded in those who have pre-existing cardiovascular conditions. How to diagnose the COVID-19 infection? The main mode of diagnosis is via the real-time PCR, where kits were synthesized and manufactured for this virus. Chest X-ray does not really show any specific symptoms, so is non-specific in diagnosis of COVID-19. The lung CT is considered a very good tool for the diagnosis of a COVID-19 infection in the lungs, and it is seen as a ground glass appearance. If you can see here, the ground glass appearance seen here, and that what you see in the lung CT, and that represents pneumonia or infection in the lung parenchyma. Maybe the most sensitive of the diagnostic tools is the CT. Nowadays, CT, it is highly sensitive, but it's not really specific for COVID-19. So any patient who presents with fever, who presents with shortness of breath, dry cough, and came from a place where there are reported cases or many reported cases of COVID-19, and we do the lung CT, and it looks like that it's mostly diagnostic. Also to confirm that with the PCR as well. So how can we prevent the COVID-19 from spreading out in the community? And if the COVID-19 spreads out in the community and it can overwhelm the healthcare sector, because the healthcare sector has a specific number of beds in the ICU or in the hospitals, and nowadays most of the countries in the world has restricted the hospital access to in the highly pandemic regions, the hospital access to the COVID-19 patients, and those who have elective surgeries or other things that can be postponed, they did postpone it. Because, for instance, in Italy, the number of patients with COVID-19 were so overwhelming to the health sector there, and as a result, they were selecting patients based on their severity of the symptoms, based on their age. So patient with very old age and very severe symptom was left on the side with minimal treatment, and they on the other side, they've given good health care for those who are of younger age and their condition is not so bad, so that the prognosis and outcome is going so that it can come better. An important factor we need to play is in flattening of the curve. As we can see, there's this curve, this blue curve, and this blue curve represents number of COVID-19 patients infected, which can become overwhelming to the healthcare sector. So this line represents how many very sick people hospitals can treat. If this number become in excess of this number here, then most probably those patients are not going to get well treated, and they are not going to be given the hospital and the medical care required. So how can we flatten the curve to look like this yellow curve here? Most of the countries in the world has most of the countries in the world had started quarantining the people infected, especially the nidus of infection in these countries, and large quarantines has taken effect in most countries in the world, including our country, Jordan, and that the primary goal for this is to prevent people from getting together and declining the number of new and emerging cases of COVID-19. So if we can decline or we can reduce the number of new infections, then we're going to keep the number of new infection below this line so that new infections can be dealt with effectively within the healthcare systems in the countries. So the first goal nowadays is prevent people from getting together, prevent gathering, prevent larger events, canceling flights, closing airports in order to flatten the curve so that the numbers of new infections decline, so that the health sector does not become overwhelmed with the number of new cases. There is no vaccine for the COVID-19. The CDC has advised people to stay at home, avoid travel and public activities, washing hands with soap and hot water, practice good respiratory hygiene in case of coughing or sneezing, covering your mouth, and in case you have a common cold or flu-like symptoms, wear a mask in order not to infect others. Avoid touching eyes, nose, or mouth with unwashed hands. So these are the preventive measures that need to be taken. What about the treatment? Is there a specific antiviral drug for treating COVID-19? No, there is no specific antiviral drug for treating COVID-19. And the main treatment aim is nowadays supportive care, where fluid and oxygen is provided. So in order to keep patients with severe conditions alive. Certain studies have shown that steroids and methylprednisolone are not recommended unless the disease is complicated by an acute respiratory distress syndrome. The ibuprofen increases the ACE2 enzyme at cell level, which could worsen COVID-19 infection. So yesterday, WHO warned against using ibuprofen for COVID-19 patients. Instead, use paracetamol. Mechanical ventilation is reserved for severe cases with COVID-19. And there are a number of experimental drugs. Scientists around the world have tried multiple drugs, let's say more than 30 drugs or drug combinations, and they have seen most effective results with, let's say, remdesivir, which is a nucleotide analog. Once used, it blocks the growing chain of the virus, or the growing chain of the RNA virus is blocked because of this nucleotide analog, so it decreases the number of newly produced viruses. Also, the lopinavir, ritonavir, which is a protease inhibitor used for HIV patients, has shown promising results when used in combination with remdesivir for treatment of COVID-19 patients. Also, chloroquine, the anti-malaria drug, has shown promising results, but all these drugs are still experimental. The patients are going clinical trials in China, in Italy, and still none of these are FDA approved for use in COVID-19 infected patients. What about the prognosis? What about the prognosis for patients with COVID-19? The mortality rate was seen to be very little until we get into 50 years of age. So after 50 years of age, we can see that the mortality rate is around 1%. It increases to around 4% after the age of 60, 8% after the age of 70, and 15% after the age of 80. So all people with pre-existing conditions of the lung, the heart, cancer, immunosuppression, on immunosuppressive therapy are more prone to infection, and more prone to death as a result of infection. Also, this figure shows that hypertension, diabetes, cardiovascular disease, respiratory disease, cancer can increase the mortality rate in these patients compared to the persons who do not have any comorbid condition. In this curve, we can see that this case represents the number of new cases which has emerged or recorded in China. So this is specifically for China, and we can see that the peak of new cases was seen around mid-February, and then the Chinese government has installed very strict measures to contain the virus, quarantined large areas of the country in millions, 50, 60, 70 millions in the Wuhan and the Hubei province. And as a result of these control measures, we can see that the number of new cases has declined. And yesterday, China has reported around 15 new cases only. And the recovery, we can see that most patients are recovering from this infection. China has recorded more than 80,000. Nowadays, more than 60,000 has recovered as a result of the illness. And we're talking about less than 10,000 which are in the final stages and in the recovery stages of the illness, but they have recorded around more than 3,000 fatalities as a result of the infection. What's the possible outcome of COVID-19 infection? This pandemic may infect most of the population, and when we say most of the population, 60 to 70% of the Earth's population might become infected. And when we say infected, this is not to spread panic, but it is like influenza virus. In order for humans to get herd immunity, they need to be infected with the virus. And once again, as influenza virus, most of the COVID-19 infections are subclinical infections, and only a small minority, we're talking about 10 to 15% of those who show symptoms, and most of those who show symptoms have mild symptoms. Only a small percentage have severe symptoms. So the infection needs to spread globally and affect more than 50% of the Earth population in order for them to have a level of immunity, so that any subsequent infections in the coming year would be the population would be immunized against it, and it would come as a common cold. Mortality will be seen in small proportion. The number of fatalities are increasing nowadays. The number of new cases are increasing, especially in Europe, but hopefully they will not exceed the 4 or 5% figures. As I said, this 4 or 5%, they are not absolute figures. We're talking about mortality rate in the sense of those who have symptoms and have been diagnosed with real-time PCR. So the actual mortality rate is less than this 5%. And scientists are working nowadays on the production of vaccine against COVID-19. But this vaccine production has been put on the fast track, and hopefully within the coming 4 to 6 months we would have this vaccine available. It's great if we can have this vaccine available before the next fall and winter season, so that people can become vaccinated against it, so that we do not see the this level of infection in the next year, or in the next fall and winter months. So to summarize, COVID-19 is a disease with high transmission but relatively low mortality, and most infections are mild or subclinical. It's spread by droplets and contact routes, mostly by the contact. Patients primarily develop fever and respiratory symptoms. Most they recover on their own, and they require few medical attention or care. Masks are advised for sick patients and close contacts, caregivers only. Frequent hand hygiene and cough etiquette should be practiced by everyone. Close contact with cases and gatherings should be avoided, especially by the vulnerable population, who are the elderly and those who have underlying conditions. Vaccine and treatment may be available within the few coming months.